Extracellular histones have been detected outside of the central nervous system, and that is in damage to organs where dying cells release histones, and they can have deleterious effects. In this study, we were interested in understanding if histones are indeed released in the injured CNS, and if so, what were some of the effects that it could have, particularly on axonal regeneration. So, in order to do this, we first obtained human CSF fluid from patients with spinal cord injury. And these were patients that had an acute injury. The CSF was collected within 24 hours. They had thoracic injury with an ASIA grade score of A. And we compared it to patients who have no injury and an ASIA grade score of E. When we probed for histone H3, we observed that histones are indeed being released in the CSF of patients with spinal cord injury. So now that we knew that histones were being released, we wanted to have an understanding what they were having specifically on the neurites. So we set up several neurite outgrowth assays. We're showing here microfluidic chambers where we have seeded cortical neurons that put out neurites across microgrooves, and they grow into another chamber. Since histones are highly charged proteins, we use a control protein, aproteinin, which has a similar isoelectric point, similar size, but no sequence homology. And what we observed is that aproteinin has no effect on neurite outgrowth. However, when we applied histones to the neurites, in a dose-dependent fashion, we observed that neurite outgrowth was being inhibited. Activated protein C, which is an endogenous protein of our coagulation system, can cleave histones and render them inactive. When we apply APC in our microfluidic chambers, we see it has no effect on neurite outgrowth. However, if we combine APC with histones, we see that it can block the inhibitory action of the histones and that you can detect long neurites. So our next question was, can we block the histone effect in vivo? So we set up optic nerve crushes. These are optic nerves that we crushed, and we labeled the regenerating axons with cholera toxin B. What we observed in the control is that the axons do grow to the injury site where they are abruptly halted. When we apply APC, and this is applied in gel foam, directly over the injury site where the histones are being released, we see that APC can promote some modest axonal regeneration. To understand the molecular actions that histones could be having on the cortical neurons, we looked for a transcription factor called YB1. And we were looking at phosphorylation of it in the presence of either mixed histones or recombinant H3 or H4. And what we observed in a dose-dependent fashion is that histones do upregulate the phosphorylation of YB1. When we combined APC with histones, we observe that we can downregulate this phosphorylation. So just to give you a graphical abstract overview, what we're showing here is that if you have injury to the CNS, dying cells at the injury sites, could be glial cells or other cell types, they release histones. And these histones are inhibitory. However, if we apply APC to the injury site, it can cleave the histones and allow some axonal regeneration to take place. I just want to end this by pointing out that I initiated these studies as a postdoc in the lab of the late Mary T. Philbin, and we would like to dedicate this paper in memory of her. Thank you.